So this is a quick review of the major concepts we've discussed up to this point in the semester. These concepts will be tested on the midterm exam, which is 50 multiple choice questions, and it covers the 5 units that we've covered so far this semester, starting with the organic molecules and ending with the nervous system. So within the organic molecules unit, some of the big picture items we looked at are just the basic functions of proteins, carbohydrates, lipids, DNA, RNA. For example, if we look at proteins, that's the major structural molecule that we see in the body. So they're very important for antibody production, important for structural strength. Many hormones are made of proteins. Enzymes are proteins. So a very, very big role for structure and function in the body when we think of proteins. Remember, the building block of proteins are amino acids that are bonded by peptide bonds. Carbohydrates, those are the major nutrient sources for most of our cells in the body. So carbohydrates start out with the monosaccharides, and then the disaccharides combine together to form polysaccharides. And again, mostly for storage of excess energy and release of energy during times when calories are low. Lipids, major functions for lipids are insulation and also energy storage. We talked about the different types of lipids. We'll talk about that shortly. And then DNA, remember, holds the genetic code. It makes up chromosomes. It has this particular structure here. It's a double-stranded nucleotide made up of nucleotides, double-stranded. And remember, a nucleotide is the repeating unit of a DNA molecule, and it's made up of sugar and phosphates forming the sides of the DNA molecule. And then we have the different nitrogen bases forming the inside rungs of the DNA molecule, holding the two sides together. So remember, DNA contains cytosine, guanine, adenine, and thymine. Recall that adenine always bonds with thymine and cytosine always bonds with guanine when we look at DNA replication. RNA is single-stranded, contains most of the nitrogen bases we see in DNA, except for instead of thymine, RNA includes uracil. And RNA, remember, participates in transcription and translation. We have messenger RNA for transcription and transfer RNA for translation. And DNA is found in the nucleus, stays in the nucleus, and again forms chromosomes and comprises what we call the genetic code. So different types of carbohydrates, like I said, when we look at that, we're looking at those repeating units. So we have one single sugar unit, we call that a monosaccharide, and that's what glucose is. That's the simplest form of energy that our cells can use. And then if we bond these glucose molecules together, we have different storage polysaccharides, which are complex molecules of monosaccharides. So they're just many strands of monosaccharides. So cellulose we find in the cell walls of plants. It gives them structure. Starch is the storage molecule we find in plants. We find that primarily in the roots. And glycogen is found in liver and muscle cells of human beings. This is how we store excess glucose. We store it as glycogen, again, in liver and muscle cells. And chitin is a polysaccharide we see in insects. It forms the hard outer shell of insects. Looking at the different types of lipids we've discussed this semester, there's 2 major categories, the steroids and the triglycerides. Remember that fats are made up of fatty acids and glycerol. So steroids, on the other hand, are made up of cholesterol. That's their basic functional molecule from which we get steroids. So all steroids contain cholesterol. Examples of steroids are the sex hormones, estrogen, testosterone, progesterone. Those are all steroids. We also talked about aldosterone. Vitamin D is a fat-soluble vitamin. Those are all examples of steroids. We looked at triglycerides. This is a triglyceride. This is what is found in body fat. So when we eat fatty meats, we're eating triglycerides of that animal, and the fat we see under our skin is also triglycerides. We talked about phospholipids, another type of lipid. These we find in the cell membrane, but these are they repel water. They have a hydrophilic side and a hydrophobic side. So the fatty acid tails repel water. That makes the inner core of the cell membrane resistant to water-soluble molecules. and the hydrophilic side, the phospholipid, the phosphate side, attracts water, so that attracts things to the surface. And remember, cholesterol is also found in the plasma membrane, and it gives stiffness to this phospholipid bilayer that forms the plasma membrane. So it keeps our cell membranes intact, that cholesterol working together with the phospholipid molecules in the cell membrane. We talked about enzymes. Excuse me. Enzymes are proteins, and they lower the activation energy for a chemical reaction to occur. For example, if we have this molecule that is a complex molecule and we want to break it down into two smaller molecules, an enzyme is required to do that. It provides the energy for this reaction to occur. So there's specific receptor sites on an enzyme for that molecule to bind. We call this the active site. And when that relationship is matched between the substrate, the chemical being acted upon, and the enzyme, we see a chemical reaction occur and new products are formed, and the enzyme does not change. So we do not need high concentrations of enzymes as long as they are specific to that reaction and they have the correct active site for that molecule. We talked about acids and bases and the pH scale. Remember that the pH is based on the number of hydrogen ions present in solution. So the lower the pH, the more hydrogen ions. So hydrogen ion concentration is a reflection of pH. So a strong acid will have a low pH. Bases, on the other hand, are a reflection of low hydrogen ion or increased hydroxide ion, which is OH minus. So the more hydroxide ion we have, the more alkaline or basic a substance is. So it'll have a higher pH. So hydrogen ions and hydroxide ions are inversely related, which means if we have high hydrogen ions in a solution, we're going to have low hydroxide ions, and that substance is an acid. And if we have a high amount of hydroxide ions, which again is OH minus, then we're going to have a low number of hydrogen ions, and that substance is a base. Then we talked about metabolism, and we talked about the basic definition of metabolism are all those chemical reactions in the body that are either breaking down or building new. So we talked about anabolism is the building of larger molecules from smaller molecules, and catabolism is the breaking down of larger molecules into smaller molecules. Both of these reactions involve either the formation or the breakdown of ATP. It requires nutrient molecules to provide the basic building blocks, and there's also waste products given off in this process, as well as heat, as some of the energy from these molecules is not completely converted into the new molecule. So we see a little bit of heat loss occurring with metabolism. But ATP is the main focus of metabolism, either breaking down ATP or creating ATP to fuel future reactions. So the equation that we see in our cells that drives metabolism is taking glucose, which is C6H12O6, in the presence of oxygen, breaking it down into carbon dioxide, water, some free electrons, and 36 to 38 ATP. Remember that this reaction occurs in the cells of our body and in the cytoplasm and mitochondria of our cells. And a lot of that excess energy is not completely converted to ATP. Much of it is given off as heat. So our body temperature is a result of the metabolism of glucose and some of that waste heat that's given off in the process. So if we look at where these reactions occur, we have three reactions occur that are part of metabolism. We have glycolysis, that is the breaking down of sugar in the cytoplasm. And at the end of glycolysis, we have 2 ATP formed and 2 pyruvic acid molecules. If there is no oxygen present, those pyruvic acid molecules will be converted into lactic acid, or if it's a yeast cell, the pyruvic acid will be converted to ethanol or alcohol. So that's when oxygen is not present. We call that anaerobic metabolism. If oxygen is present, recall that pyruvate enters the mitochondria and is broken down into, or converted, I should say, to acetyl coenzyme A. Then it enters the Krebs cycle, where 2 ATP are produced, and then if there's still oxygen present, it enters the electron transport chain, where we can get up to 34 to 36 ATP produced. Remember that The fluid inside the mitochondria is called the mitochondrial matrix. That's where the Krebs cycle occurs. And the electron transport chain occurs in the membrane of the mitochondria called the cristae. So what are the reactants and products of each of these processes? Well, we know with glucose and glycolysis we get 2 molecules of pyruvate and 2 ATP. If we have oxygen present and that acetyl coenzyme A is formed, enters the Krebs cycle, the waste product we see of the Krebs cycle is carbon dioxide, and we also get 2 ATP. And lots of enzymes are created in the process of the Krebs cycle. So we have many enzymes. We have the coenzymes of NAD plus and FAD plus that are converted into, NADH is converted to NAD plus and FADH2 to FAD. So we see that conversion, that interconversion occurring between the Krebs cycle and the electron transport chain. In this case, they call it oxidative phosphorylation, but we'll just call that the electron transport chain. So what we see is carbon dioxide is given off and 2 ATP in the Krebs cycle. Lots of enzymes. We have these coenzyme carriers of electrons that are formed, and then they are released to the molecules in electron transport chain, and we see a release of water in the process of the electron transport chain, and lots of ATP, like I said, 32 to 34, or even up as high as 36 ATP in the electron transport chain. So special reactions that are part of each that you should be aware of. Again, remember the Krebs cycle is a long process. There's 2 cycles of the Krebs per pyruvic acid molecule. Remember that there's 2 pyruvic acid molecules that are formed from every glucose. So because of that, we see 2 cycles of the Krebs per glucose molecule, and many steps involved as we have these intermediate molecules that are formed along the way. The electron transport chain has those special proteins embedded in the matrix of the membrane, and we see the ATP synthase pump in the electron transport chain, and that pumps hydrogen ions. Well, hydrogen ions are pumped out of the cell or out of the mitochondria, and then they flow back in and they fuel that ATP synthase pump. So this is described in chapter 24 of your textbook, looking at the different diagrams for the electron transport chain and the Krebs cycle. So you might want to review those, but again, we're not going to go into great detail. This is a midterm exam. We're just looking at the basics. So carbon dioxide is produced in the Krebs, water is produced in the electron transport chain, pyruvic acid from glycolysis, and the Krebs cycle involves many enzymes and intermediate molecules, and the electron transport chain uses proteins embedded in the matrix as well as the ATP synthase pump. So other ways that we can manufacture or store glucose, we talked about some terms. We talked about how the liver can use substances like fat or protein to make sugar. So we call that gluconeogenesis when we're making glucose from non-glucose molecules like protein or fat. The liver can also break down glycogen and release glucose to the blood, so we call that glycogenolysis. Remember, lysis means to split, so if we split glycogen, we are releasing glucose. We can absorb glucose from the intestines when we eat, and then we can also break down fat, called in the process of lipolysis, releasing free fatty acids, again, which can be used by the liver to make new glucose via gluconeogenesis. So these are some of those reactions we talked about this semester that can help us release glucose into the blood. Then we talked about the endocrine system. We talked about how different hormones act on the cell. We have steroid hormones and peptide hormones. So recall that peptide hormones are water-soluble hormones, so they cannot pass through the cell. So the way that peptide hormones act on their target cells is they bind to receptors on the cell surface, and then they activate some other reaction inside the cell to occur. But those hormones cannot exert their effects inside the cell. They can only bind to receptors on the surface because they are water soluble. So hormones like insulin, for example, can only act on the outside on those surface receptors. Steroid hormones like estrogen, progesterone, testosterone, aldosterone, vitamin D, those can diffuse right through that fatty membrane because they are also fat soluble, steroid hormones are. So as a result, they can go through the membrane and enter through the nuclear pores into the nucleus and alter DNA and cause new production of a substance by that target cell. So we talked about how peptide hormones are kind of like a fast food restaurant where you can drive up and the molecules or what you need is readily available through in secretory vesicles because you just bind to the receptor and the substance you're looking for from that target cell may be readily available, where the steroid hormones are kind of like a sit-down restaurant. You go in, you order, you change the DNA, and it takes a while for that change to occur. So when we look at hormones, how long they last, how long it takes for them to enter the blood, we see that these stay in the blood a little bit longer compared to peptide hormones. It takes a little longer. Steroid hormones take a little longer to be produced. I should say what is produced by the target cell from a steroid hormone takes a little bit longer. And we look at the relationship between the hypothalamus and the pituitary gland. Remember, the pituitary gland is the master gland of the endocrine system, but it is tightly controlled by the hypothalamus, which is part of the brain, part of the central nervous system. So we have the anterior pituitary and the posterior pituitary. The anterior pituitary is a true gland. It releases FSH and LH, growth hormone, adrenocorticotropic hormone, which is ACTH, prolactin, those tropic hormones, TSH, thyroid stimulating hormone, those are all produced by the anterior pituitary, and they many of them are tropic hormones. The only one that is not tropic is growth hormone and prolactin. Those do not act on other endocrine glands telling them to secrete their hormones. Remember, the posterior pituitary is just an extension of the hypothalamus. It's not a true endocrine gland. The actual hormones are made up in the hypothalamus and stored in the posterior pituitary. And the two hormones of the posterior pituitary are oxytocin and ADH. How we stimulate endocrine glands to do their job, there's 3 ways that we stimulate them. Most commonly, they're stimulated by some hormone from the anterior pituitary. For example, the thyroid gland secretes T3 and T4 under the influence of TSH from the anterior pituitary. So that's passing through the blood to those target organs, which are endocrine glands. The adrenal cortex secretes cortisol under the direction of ACTH from the anterior pituitary. The gonads, the testes, secretes testosterone under the influence of LH. Remember, LH is the hormone from the anterior pituitary that promotes testosterone secretion. So that's the specific hormone that stimulates testosterone. And remember, LH in the female stimulates ovulation. Humoral or blood-based means there's some chemical in the blood that is stimulating an endocrine gland. So when here we see the parathyroid glands near the thyroid, when they detect changes in calcium from the blood, for example, low blood calcium, then the parathyroid glands secrete parathyroid hormone. So the stimulus is something in the blood. And then only neural stimulus we see of an endocrine gland is that sympathetic neuron that goes to the adrenal medulla, causing the adrenal medulla to secrete norepinephrine to the blood. So here a neuron is stimulating this endocrine organ. Here something in the blood is stimulating the endocrine organ, and here a hormone is acting on those endocrine organs. So there's hormonal, humoral, and neural stimulation of endocrine organs. Where hormones are secreted, remember they're always secreted to the blood. How hormones are regulated, the hypothalamus is constantly sampling the environment and determining levels of hormones, and then it will influence the anterior pituitary if we need more hormones to be produced. For example, we talked about these different hormones from the anterior pituitary. They're secreted to the blood. But remember, there's this special portal system between the hypothalamus and the anterior pituitary. This picture is opposite of the previous picture that's flipped around. So the front of the brain is actually over here. So this is the anterior, this is the posterior. This other picture here is the reverse. So this is the anterior, this is the posterior. Be sure you don't get confused by the pictures that are oriented 2 different directions. But anyway, these are all the hormones from the anterior pituitary. So there's this special portal that's called the hypophyseal portal system that communicates only between the hypothalamus and the anterior pituitary. So only the hormones from the hypothalamus, like GnRH, can go from the hypothalamus to the anterior pituitary through these special portal vessels that are not in direct communication with the general blood supply. We talked about upregulation and downregulation. If we have excess hormone, what we might find is over time the receptors on that target cell might decrease, so there's less influence of that excess on those target cells. Upregulation, if we don't have enough of a particular hormone, we might see an increase in receptors over time, so we call that upregulation. So the up or the down just refers to if we're adding or removing receptors for a particular hormone on a target cell. Then we talked about the reproductive system. The reproductive system, we talked about how sperm and egg are formed in lab. We looked at the structures in the male and female reproductive tract, quite a few different processes here. So let's look at gametogenesis. Remember, we talked about mitosis and meiosis, and spermatogenesis and oogenesis. Remember, spermatogenesis occurs throughout a man's life. Oogenesis occurs before birth, but the process doesn't complete until later in life when the egg is fertilized. So remember that we have meiosis 1 and meiosis 2. So we start out with a primary spermatocyte and a primary oocyte. And then when meiosis 1 is complete, we have 2 haploid cells. And in the female, we have a polar body and a haploid secondary oocyte. So after meiosis 1, we have secondary spermatocytes and secondary oocytes. After meiosis 2, we have spermatids, still haploid, and we have, again, this ovulated secondary oocyte, but it doesn't become an ovum and complete meiosis 2 until fertilized by the sperm. Then it becomes an ovum. So it's important to remember that we don't complete meiosis 2 until after that egg has been fertilized. Then it's called an ovum. So that's when meiosis 2 is complete. So remember, at the end we have 4 haploid cells at the end of meiosis. The same thing in oogenesis, but we just have polar bodies, remember, that are formed. So we would have 1 mature ovum that's been fertilized, and then up to 3 polar bodies, all haploid. So the process of becoming haploid occurs after meiosis 1. And the special events, remember, we have crossing over, we have random assortment, independent assortment that leads to genetic variation in these cells. So these cells are all carrying different alleles from the parents because of the prophase 1 of meiosis 1, where we see that crossing over occurring of those homologous chromosomes. So where other structures we can talk about, remember the structures in the ovary, specifically the corpus luteum is the remaining follicle cells that remain in the ovary, and they secrete progesterone. And remember, in the male, the structures in the testes, we have the seminiferous tubules, and the epididymis is where sperm are formed. So sperm are formed in the seminiferous tubules with the help of these Sertoli cells. Remember, they surround the developing sperm cells and are involved in nurturing and promoting the development of the spermatids that will enter the lumen and leave the seminiferous tubules, enter the epididymis, and then enter the rest of the male reproductive tract. Remember, semen is made up of secretions from the different ducts of the male reproductive tract. So semen comes from the prostate, a little bit of the secretions come from the seminiferous tubules, from the prostate, the seminal vesicles, and the bulbourethral glands all contribute semen that nourish and support the sperm. Again, the corpus luteum in the female is going to remain, and if fertilization took place and a zygote was formed, remember, sperm and egg coming together forms a zygote. If a zygote is formed, that zygote will develop. We talked about the blastula and the morula, and as that blastocyst implants in the endometrium of the uterus, it starts to secrete a hormone called HCG, and that HCG is what is detected in home pregnancy kits. And again, looking at the chromosome number, we have 23 chromosomes after meiosis 1 is complete. All the N cells here are all 23 chromosomes. Prior to that, spermatogonium, primary spermatocyte, remember those had 46 chromosomes. They were diploid. They had two pairs of, or a pair of chromosomes rather than one single set. Then we talked about the nervous system. We have the central nervous system, which is made up of the brain and spinal cord, and then we have the peripheral nervous system, which is made up of the cranial nerves, of which we have 12 pair, and spinal nerves, of which we have 31 pair. And remember, the role of the nervous system is to conduct impulses from the brain to the peripheral nervous system and back again. Those impulses are called action potentials and graded potentials. I'll move this down to the next page. So if we look at resting membrane potential of a nerve cell is negative 90. We see some stimulation that can occur. Anytime we see any stimulation that is below the resting membrane potential or a decrease, we would call that an inhibitory postsynaptic potential because it's going further from threshold. And if we look at any stimulus that rises the resting membrane potential, we would call that an excitatory postsynaptic potential. But those are all graded potentials. They don't quite reach threshold. So in this example here, we have a cell that's just kind of hanging out here this is a little lower of a resting membrane, or a little higher of a resting membrane potential. This is negative 90, so we could say maybe negative 70 the cell is hanging out at. But either way, when we go further away from resting membrane potential, it's an inhibitory potential, and if it's higher up, it's an excitatory, and these can add together. So if I added these five here together, I see more going in the down direction. If I added up how far down they were and compared how the amplitude of these two, I would see we would tend to be a little inhibitory. But in this case, some stimulus occurred over time that caused this potential to rise up rapidly and reach threshold. Once we reach threshold, we have an action potential. And this is the potential that's going to be delivered to the brain or the spinal cord for processing. These would not make it to the brain or spinal cord for processing because they did not reach threshold. So when we talk about this resting membrane potential, in this case it looks like it's about negative 60, I would say. What contributes to that? Remember, we have leak channels for sodium and potassium, and we also have the sodium-potassium pump. And if we think about the importance of these channels, the sodium-potassium pump is really critical because it takes the ions and puts them in opposite directions according to their concentration gradient. So it maintains a concentration gradient so we see the flow of potassium and sodium. If we didn't have the pump, we wouldn't have flow through leak channels. So as a result, the sodium-potassium pump really plays a critical role in establishing that resting membrane potential. And then we also have the help of these negatively charged proteins inside the cell as well. We talked about neurotransmitters and their effects on a target cell. I'll move that down as well. So neurotransmitters are held in the axon terminals of neurons, and they are released when there's a stimulus, might be an action potential coming down the neuron. But regardless, we have these neurotransmitters held in these synaptic vesicles, and they bind to receptors on the postganglionic neuron. Or this could be a cardiac muscle, smooth muscle, or gland. Regardless, neurotransmitters released from the presynaptic membrane are going to diffuse across the membrane and bind to these receptors on the postsynaptic membrane. So it's important that we recognize that these neurotransmitters, if it's a sympathetic neuron, would be norepinephrine, and if it's a parasympathetic neuron, it would be acetylcholine, and this would be a target cell in this case. But if we're looking at the two neurons in series that form the autonomic nervous system, then if this was the preganglionic neuron, this would be acetylcholine in both the sympathetic and the parasympathetic nervous systems. I'm not going to test you a lot on the autonomic nervous system. We haven't gone into that in a great in great depth yet, so be more general in your understanding of the autonomic nervous system. You do not have to know about specific receptors for this exam, like nicotinic, muscarinic. You don't need to know those receptors for this midterm exam. Again, the midterm exam is very general in nature, just having you understand basic information related to the different systems or units. So we talked about sensory and motor neurons. So you should know that the sensory neurons come in via the dorsal root and synapse on the dorsal horn, and then they, if it's a reflex, if it's an autonomic reflex as shown here, there'll be an interneuron that will connect that impulse and carry it right out to the response, which comes out the ventral horn. So we see the motor response is through the ventral horn, and then it synapses out on this organ. So this is autonomic. This is smooth muscle of the digestive tract. So we see the same thing dealing with skeletal muscle. This is the somatic reflex. So we have a sensory neuron coming in through the dorsal root, and then the motor neuron coming out through the ventral root to cause some voluntary response here. So the ventral root, remember, contains the response by the body, and the dorsal root contains sensory information that's coming into the central nervous system. So we talked about the different tracts for perceiving sensation. I'm not going to hold you in great detail of where they cross over, where the first, second, and third order neurons are. That's all very detailed information that's left for a unit exam. For the midterm exam, I would just like you to know that there are 3 sensory tracts, and that they come up and synapse on the primary somatosensory cortex. But you don't have to know details within each tract. Just basically know that when we talk about tracts for sensation, we have the spinal cerebellar that gives unconscious proprioception, and the spinal thalamic, which tells us about temperature and pain sensations. Those are really critical sensations that we need so we can keep our body safe. So that's the spinal thalamic ascending tract. And then the descending tract, we have the motor cortex. Remember, we have the motor cortex, which is that postcentral gyrus up here in the brain. That comes down the spinal cord, and that's the upper motor neuron. We have the lower motor neuron synapsing on skeletal muscle. So these descending tracts are the corticospinal tracts, which control voluntary movement. So the only thing you need to know is that it's the corticospinal tract is a descending tract serving skeletal muscle. We also call those pyramidal tracts as well, the controlling voluntary skeletal muscle. And then we have the autonomic nervous system. The sympathetic, remember, relies on norepinephrine. The parasympathetic relies on acetylcholine acting on these target organs. And just know the basic functions. For example, we see in the heart increased stroke volume, which is just increased force of contraction and heart rate. We see vasoconstriction throughout the body. Reduced digestion, secretion, inhibiting bladder contraction, and then the sympathetic effect. So this is just a real general kind of overview of the autonomic nervous system, and that's all you need to know for the midterm exam. You don't need to know great details that we're studying right now for our unit exam. That'll be much more detailed. So if you have specific questions about the midterm exam, feel free to email me. Again, it's 50 questions, and if you review the pictures and the content on this review sheet, I think you'll be in good shape for the midterm exam.